Okay, so we have heard from Dr. David Simon, so I'm not going to introduce him again, but Dr. Tim Greenamyre is the Love Family Professor and Director of the Pittsburgh Institute for Neurodegenerative Diseases. He's also this year's winner of the Jay Van Andel Award for Outstanding Achievement in Parkinson's Disease Research. So, yeah, thank you so much for coming, and please take it away.
Yeah, thank you. I'll just start by saying, yeah, that was a very brief introduction to what could be a very long list of awards and accomplishments, some of which we'll hear about today. I think that the conference, and it's an honor personally to get a chance to interview Tim because, well, one, his work has influenced mine, and not just mine. I mean, many people's. You can't really talk about mitochondrial dysfunction and Parkinson's and not come back to his work, and his work has influenced beyond that, for example, and what I do as well. And we'll talk about some of that. But at the beginning, maybe you want to introduce yourself to the audience.
I'm Tim Greenamyre. I am a neurologist and neuroscientist. I started out not knowing what I wanted to do. I ended up getting my PhD in a laboratory of a role model named Ann Young that some of you may have heard of, who was a pioneer in basal ganglia function. Basal ganglia are the brain areas that help control movement. She took care of Parkinson's patients and Huntington's patients and was a role model for me. When I went to University of Michigan, I got my PhD with her, and then did a residency in neurology, specializing in movement disorders. And I've been practicing neurology, taking care of movement patients since 1990 or so. I have a lab and spend most of my time in the lab and part of my time seeing patients. So that's how I divide my time, what occupies my efforts.
And what was your initial inspiration, your reasons for choosing to focus your career on Parkinson's?
I really liked the patients, the people who have Parkinson's disease. I just made a connection with them right away. And I liked the fact that we could help them and work on ways to better help them in our research.
And then you told us a little bit about the path and how you got started on that. But I don't know if you want to say a little more on what got you down the path for the research that we'll talk about in a minute.
Yeah, when I was finishing my residency, I was writing my first grant application. I knew that I was going to the University of Rochester for my first job, and was thinking about my first research project that would be funded hopefully by the NIH or a Parkinson's related foundation. It was an exciting time for Parkinson's research. There was new evidence that the mitochondria might not be working in Parkinson's disease properly, as you heard from David just recently. There was also evidence that pesticides used in agriculture may predispose people to Parkinson's disease. And I thought of ways that I might connect those two things and work on a compound or a component of the mitochondria called complex 1, and see if I could figure out what was wrong with complex 1 in Parkinson's disease. And to do that, we eventually gave a complex 1 active toxin to rats and reproduced lots of the features of Parkinson's disease in the rats. So it was just a very beginning of a career that has gone from that model to many other aspects of Parkinson's disease over the years.
So you may have touched on this there, but what do you consider, I mean, you're known for a lot of different things that you've done, but what do you consider your first major achievement? Not necessarily the one that got the most attention, but what you consider the first time you did something in the lab and came up with a result that you said, this is really important.
I think when we gave rotenone, let me explain what rotenone is. Rotenone is a natural compound. It comes from plants of different species. It was used as a fish toxin for centuries in South America. The natives would put rotenone into the lakes and kill fish as a way to harvest the fish. It's a complex 1 inhibitor. It blocks the actions of complex 1 and causes mitochondrial impairment. And I was trying to reproduce the complex 1 defect that people had already described in Parkinson's disease. And so we took rotenone, gave it to rats, and found a dose that made the rats Parkinsonian, caused the same kind of loss of the dopamine neurons, caused the same kind of synuclein aggregates and Lewy pathology that are seen in patients. And it was really a good model of Parkinson's disease from many aspects. It led to the idea that mitochondrial function is important in Parkinson's disease. It led to new models for testing therapeutics. And I think it's been cited in the literature about 5,000 times. So people that are very in tune to the idea that rotenone can inhibit complex 1 and cause Parkinson's disease. I think that first paper that we published on this was probably the biggest accomplishment.
And with that in context, about 5,000 citations for a single paper like that is in a good measure, single measure of huge impact on that. You know, it's influenced many people's work. But I touched on this in my talk, but I don't know if you want to explain why, you know, rotenone, sorry, MPTP had been discovered before, also a mitochondrial toxin, but the rotenone story was different. What did that tell us that was different from the MPTP story?
Yeah, so the MPTP story, are you all familiar with that?
I mentioned it, but
okay. So MPTP, when it gets into the bloodstream, can get into the brain, and it gets into dopamine neurons because it acts, the dopamine, when dopamine is released from a dopamine nerve cell, it gets taken back up into that cell so that dopamine can be reused. And the way it gets taken up into those dopamine cells is through something called the dopamine transporter. The ability of MPTP to selectively cause degeneration in the dopamine neurons was because the toxin MPTP is a substrate for the dopamine transporter, meaning that it goes in through the dopamine transporter, and that's how it is very selective for those neurons. Rotenone, on the other hand, gets in every cell, gets in all of the neurons. And so the MPTP model tested the idea that if you inhibit complex 1 in dopamine cells, you kill dopamine cells. Rotenone asked a slightly different question, that if you inhibit complex 1 in all the cells, do you still get selective dopamine degeneration? Does that make sense?
Yeah.
Okay.
Oh, great. So rotenone, I had also mentioned, was previously used as an organic pesticide.
Right.
I think it's no longer considered okay for that. Can you talk about how your work influenced that removal of it, and also I'll ask about paraquat next, but and how if you did, and how the interactions went with regulatory agencies.
I didn't have a ton of interaction with regulatory agencies, but shortly after our first paper was published, I got a call from the Australian regulatory agencies, and they wanted more information because they were considering banning it. I also got a call directly on my office line. I have no idea how this person got my direct office line. But it was a person screaming at me because they were the sole importer of rotenone into the United States, and I was ruining their business. I think that rotenone has been banned for most purposes in the US, except is still used in lakes and reservoirs to kill nuisance fish or to sample fish populations. So it's used in waterways that we derive drinking water from. It's assumed that the rotenone that they use detoxifies rapidly, but I'm not sure that that's the case. I think that rotenone persists for longer than people anticipated.
And paraquat, you want to talk about paraquat?
Yeah, sure. Paraquat is an herbicide, a weed killer that is extremely toxic. It's so toxic that it is commonly used to commit suicide. It is used in agriculture to kill all the weeds before you plant your crops. And it's used in all kinds of agriculture, including viniculture, vineyards. So paraquat is all over the place. The people who are most susceptible are the people who apply the paraquat. It's a fact that paraquat enters the human body no matter what protective gear you wear. There are studies that were hidden by the company that makes paraquat that farmers who were tested wearing essentially a hazmat suit still got paraquat in their systems. And paraquat, once it's in the brain, there's no known way that it gets out of the brain. It persists forever, and it causes progressive oxidative damage, eventually killing the dopamine neurons. Paraquat has been under litigation for a number of years. I participated in some of the litigation. It's a very interesting read to go and read something called the Paraquat Papers. You can find it online. It describes how Syngenta, the manufacturer of paraquat, hid data that paraquat is neurotoxic, that it can cause Parkinson's disease. They have generally been obstructive and deceptive in their interactions with the public. I lost my train of thought.
The paraquat papers.
Yeah, it's gone.
Okay, but you've said a lot on that, and I guess so why is it that regulatory agencies haven't banned paraquat? You just gave a lot of damning evidence.
Yeah, there's a fundamental difference between the regulatory agencies in the United States versus those in most of the world. In most of the world, if you introduce a new chemical, you have to show that it's safe. In the United States, in the EPA, you have to prove that something is very toxic before you can ban the compound. So it's sort of backwards. I'm sorry.
Hey, what about the dosing in the, you've mentioned about the dosing in animals versus humans.
Thank you. There's abundant evidence that if you give paraquat to an animal, it will cause nigrostriatal degeneration, the degeneration of the dopamine neurons that die in Parkinson's disease. Of course, you do this in experimental animals such as rats or mice. You don't give it in the same way that humans are exposed. You typically inject it into the animals. Humans get it through inhalation and ingestion when they're applying the compounds. The EPA doesn't consider it relevant data if it's not administered in exactly the same way at the same doses that apply in humans. Of course, you can't apply the same standards to rats and expect to wait 20 years for somebody to get, for the rats to get Parkinson's disease. You have to give higher doses, so you sort of trade the time necessary to create the disease. In rats, you trade time for a dose. You use a higher dose to make it happen more rapidly. And there was a huge body of literature showing that paraquat causes Parkinson's like features in experimental animals. There's huge epidemiologic evidence supporting this, and it has a mechanism of action of causing oxidative damage that is known to occur in Parkinson's disease. All of these things argue strongly against the fact that paraquat should be used in agriculture. The EPA also sabotaged an appointment of an expert in paraquat in animals. She was going to be appointed to their council to evaluate the toxicity and appropriateness of paraquat. They sabotaged her appointment so that she didn't participate in these meetings, and the outcome is that paraquat is still in the market. Paraquat has been banned throughout Europe. It's banned even in China. It's manufactured in England where it can't be used. It's manufactured in China where it can't be used, and it's sent to the United States where it can be used. The amount of paraquat that's being used is increasing every year, and to an increasing extent, we're all getting exposed to it.
Wow. Actually, is Ray Dorsey talking this afternoon?
We've got. I think he's joining by Zoom, isn't he?
Oh, okay.
Also got a pre-record from Ray.
Okay, just another neurologist who I'm sure has something to say about this and related topics.
Yeah, Ray will argue that Parkinson's disease can be prevented, that it's all entirely related to environmental toxicants. I think my view is slightly different, that we can prevent a lot of Parkinson's disease, but not all of Parkinson's disease. There's genetic components. There's a lot of factors that influence your chances of getting Parkinson's disease beyond industrial toxicants.
And so, that's really interesting. I want to move on to another topic. A lot of your work and what we talked about so far relates to mitochondrial dysfunction, but you've also done work on other genetic causes like LRRK2. And can you tell us a little about what you've done that's most important related to our understanding of LRRK2?
Right. I think the most important thing I've done with LRRK2 is show that it plays a role in sporadic or idiopathic Parkinson's disease. The idea is that LRRK2, when mutated, is a cause of Parkinson's disease. That is not particularly common. It is probably less than 5% of Parkinson's disease. It is associated specifically with mutations, and there are several mutations in the LRRK2 gene that cause people to have Parkinson's disease. What we showed was, well, let me explain, that the mutations that cause Parkinson's disease cause an increase in the activity of the LRRK2 enzyme. It's called a kinase. So you have increased kinase activity when you have a mutation. People who have normal LRRK2 without the increase in that kinase activity, we showed actually do have an increase in kinase activity that's not related to the mutation. The importance of this is that pharmaceutical companies were developing LRRK2 kinase inhibitors for people who had mutations. So 3 or 4% of the population of people who have Parkinson's disease. What our data suggests is that LRRK2 is active in the sporadic cases, all the other cases that don't have LRRK2 mutations. What that means is that instead of treating 3 or 4% of the population, LRRK2 kinase inhibitors may be useful for something like 100% of Parkinson's patients. Based in part on our work that showed this, the first LRRK2 kinase inhibitor trial that's going on right now between Biogen and Denali is working at both mutation carriers and sporadic patients who don't have mutations. I'm very optimistic that there'll be some positive results, but we won't know for some time.
It's great. Those trials are going on now. There's actually another trial that probably is partly inspired by the work you just described that's selecting people who don't necessarily have a mutation, but based on peripheral measure of LRRK2 kinase activity. Although my understanding was it was only like maybe 25–30% of people who would meet their criteria. You said you think maybe almost all patients have high LRRK2.
Well, I think most patients have high LRRK2 activity. I think their algorithm for determining who is at risk is a mixture of genetic SNPs and some biochemical testing. And it's sort of a black box that they haven't disclosed.
Okay, yeah, very interesting. Thanks. So one other thing I want to ask you about, I think you've given us permission to talk about the fact that you've been diagnosed with Parkinson's. In fact, you've been very open about that. There was a really nice article in Science magazine that got a lot of press about your diagnosis. How has that influenced your the work that you do, or hasn't?
It hasn't really influenced the work that I do a lot. I've always worked with the idea of translating our findings from the laboratory and from work in rats and cells in a dish to thinking about how I can make that apply to improving Parkinson's treatment. Where it has changed is in my practice of neurology in seeing Parkinson's patients. I've been following Parkinson's patients for 35 years and always felt that I had a good feel for the symptoms that people were describing to me. And I'm getting a much refined understanding of what those symptoms actually are and what they feel like, and what different changes that you go through mean on a functional day-to-day basis. So I think I have a much better understanding of what people are telling me when I see them in the clinic, and suggesting changes in their therapy or their daily activities or so on.
Thank you. You're also have been a member of the Cure Parkinson's Linked Clinical Trials Committee for quite a while. You want to tell us about about your relationship with Cure Parkinson's and that committee and your thoughts on that.
Sure. I don't know if you're all aware of this, but the organization, International Linked Clinical Trials, and Cure Parkinson's, gets together on a yearly basis and reviews a portfolio of potential compounds or drugs that might be useful in Parkinson's disease. The committee is composed of an international group of experts in Parkinson's disease, clinical people, basic scientists, and people like me who do a little of both. Simon Stott, who is, I don't know his official position.
Director of Research. He was on screen yesterday. So even though he's 6'7" or something like that, he was in a tiny little box in the top of the screen.
Simon does due diligence and sort of surveys the field, gets suggestions, and puts together a dossier on each compound that might be useful in Parkinson's disease. Then the committee evaluates that dossier and figures out whether something is ready to go or whether it needs a little more time to cook before it's done. The result of all of this is that a number of clinical trials have been performed with compounds. It's often repurposing of drugs that are available that are approved for other purposes. The idea is that we may be able to take things off the shelf that have been used for something completely unrelated to Parkinson's disease that may be a fast track for new therapies. And I don't know how many clinical trials have been performed now, 30 something.
You're stealing my thunder on the next presentation. We say it's around about 30, but there are around sort of 15 or so clinical trials that have been completed to date. So the portfolio is sort of busy at any given time, but I'm going to elaborate a bit more on that after coffee break.
Great. So next question is sort of a follow-up on that, which is a question I get asked a lot. I'm very interested to hear what you think. Among all the things we've talked about, Linked Clinical Trials Committee or outside, what are the most promising neuroprotective strategies now that are either in clinical trials or approaching?
This is a biased view, but I'm very optimistic about LRRK2 kinase inhibitors. There is different flavors of LRRK2 inhibitors are in trial at this point, from gene therapy approaches to small molecule drugs to take. I think that targeting inflammation may be a very important goal to stop progression of the disease. I'm optimistic that some of the synuclein targeting approaches may be useful. You've probably heard that there is a synuclein antibody therapy that's been tried and is showing hints of promise. The Roche compound, Patrick Brundon has championed this, and it's showing hints of protection, of slowing the course of the disease. I think the antibody approach is likely the least effective way to treat synuclein. I think it's a very good target. I think if we can reduce synuclein levels, it will be protective. But I think there are approaches that will be more effective than trying the antibodies.
Thanks. One other follow-up regarding the LRRK2 inhibitors. So it's interesting that there's some data that people with Parkinson's who have a LRRK2 mutation on average progress slower than people who don't. And of course, variant that caused a risk of Parkinson's might be different than what influences progression. So is there concern that, well, if this variant is associated with slower progression, does it make sense to inhibit the influence of this mutation in order to slow progression further?
I think it's a theoretical question that we won't be able to answer until we do the clinical trial.
Yeah.
Personally, I'm not so concerned about it, but I think it's a theoretical risk.
I think some of your own data, among others, shows in support of the idea that it's still very promising that the inhibitors in experimental models do seem to protect.
Right.
So it's I don't know.
Do we have time for a few questions?
Oh, sure. Why don't we go do that? You have questions from the audience?
Exactly. I have a question. Sorry, jumping in first. You mentioned about the hidden paraquat papers. Were those published and then hidden from view, or what happened?
These are Syngenta internal documents.
Okay.
One of the Syngenta, or one of the paraquat papers is a report I wrote on paraquat and its relationship to Parkinson's disease and some specific individuals who were exposed to paraquat.
And that's been suppressed.
That was been suppressed. It came out in Discovery and someone leaked it to The Guardian, and it's online now, much to Syngenta's chagrin.
Yeah, I think being able to signpost that, make sure that we can try and sort of stop it. Syngenta feels important.
Right.
Yeah. They don't respond to letters, by the way. We've tried. Are there questions from the floor?
What are some common herbicides that we might see at Home Depot that use paraquat?
Paraquat isn't available for that kind of thing anymore. Paraquat is used exclusively for agriculture. You at this point in the 2000s, since probably 2004 or so, you've had to get specific training in how to apply and use paraquat. There are other pesticides that are still available. Some of the insecticides that we use and spray around the house are implicated as potential causes of Parkinson's disease. Rotenone is not available except to people who are officially using it to sample fish populations and that kind of thing. But there are other things that are household insecticides that are still out there. Rotenone, interestingly, used to be available for vegetable gardening. I used it in my tomato plants. It was a puffer, kind of a can that you would squeeze, and the powder would come out in a cloud, and you breathe that in, and it does its deleterious thing and ends up with Parkinson's disease in some cases. One of the things to recognize is that two people who get the same exposure to a particular compound may have completely different responses to that compound. One person may develop Parkinson's disease and one person may be perfectly healthy. You have in your liver the capacity to metabolize and detoxify compounds, but everybody's complement of enzymes in the liver is a little bit different. Some are more efficient than others for specific kinds of compounds, and some are less effective And it's the combination of your lifetime of exposures to different things and your genetic makeup that determines your risk of ending up with Parkinson's disease for a given exposure.
So given all these different, I guess we'd say, expressions of Parkinson's, we look around the room or among our peers with PD, we see it all looks similar. Is there any way, like telltale signs that would indicate this your rate of progression is slow, so that indicates more of this kind of causal activity, say paraquat, or maybe even more generally a gene mutation versus like we've learned, I guess there's these up to 20 different gene mutations that could possibly involve Parkinson's. Do they leave telltale signs in our experience with Parkinson's, in the look of our Parkinson's, in the progression of our Parkinson's, that would indicate its root causes?
We don't have that information except in genetic cases of the disease. So as David was saying, there's some evidence that LRRK2 mutation carriers may have a slower rate of progression, and GBA carriers may have a more rapid progression of disease. There's little bits of evidence that certain LRRK2 mutations may have a clinical picture that's a little bit different than sporadic PD, although that's not entirely clear. Regarding toxicants and exposures and the symptoms that people develop, we just don't have enough information.
Yeah, I have a question that because you've been a practicing physician, if anybody's doing work on exposure to medications, because I worked in a peds ICU for 20 years. We actually mixed our own meds when I started. Dopamine, I swam in it because that's the first line drug for hemodynamic instability in kids. Is there anything out there on that? And do you think there's any relationship to that, your practice and exposure to any of those kind of things that you may have come across?
I don't think so. First, regarding dopamine, which is used to control blood pressure in ICU patients, that dopamine doesn't really get into the brain. The reason is that there's the blood-brain barrier, and that's why we have to use L-DOPA, which becomes dopamine in the brain. Certain medications, when you're taking them, cause something that looks very much like Parkinson's disease. Some of the antipsychotic medications and some of the medicines used for nausea and GI motility, things like Reglan, can cause a picture that looks very much like Parkinson's disease. It's typically reversible. Occasionally, it's reversible when you stop the medication. Occasionally, it's permanent. There's some evidence that some of the antipsychotics can actually be slightly neurotoxic, particularly the older antipsychotics, or neuroleptics, they're sometimes called. But generally, the answer is that medicines don't cause Parkinson's disease.
Are there any more questions for Tim and for David?
Yeah, can I have one? So you have said that chemicals like paraquat, protein are out there, and for people, that there's not that much that we can do to perhaps decrease our exposure, apart from what the government, tells companies whether or not they can be used. So this is not something that people can necessarily control or even be aware of. But is there a different strategy to perhaps neutralize the effect of this exposure
after it's happened? Are there any strategies like medication, increasing their antioxidants or something like that that could, kind of, get the balance right again? This is something that has been explored.
Right, that's an important question. That question is predicated on the fact that we have to know that somebody got an exposure to something that was toxic, and usually that's not the case. There are potential ways that you could mitigate the toxicity. Again, I'm optimistic that something like a LRH2 kinase inhibitor might be protective after exposure to certain kinds of toxins. The problem really is that when you get exposed and when you get Parkinson's disease, may be separated by 20, 30 years. And so you would have to recognize that you were exposed and then start taking a drug to mitigate the possible damage. You may have to take that for a couple of decades, and it may take that long to figure out whether it's working or not. I don't think that that's anything that could be tested in a clinical trial or approved by the FDA. I think that it's a difficult problem.
Related to that, do you recommend buying organic to avoid paraquat?
Yeah, I think buying organic is good. You have to have a certain income level to afford to buy organic, and I think that's not as much as I buy organic myself. I'm privileged, and I recognize that. There are many areas of the country and inner city areas where buying organic is not a possibility for a variety of reasons.
So is paraquat linked to any other diseases so that, you could go as like an army against this?
It's associated with cancer, I believe.
Kind of, everybody in here is already affected some way, shape, or form or another with Parkinson's. Is there anybody out there addressing what some of the causes are that we can start to prevent this from going further? Now what I see is everybody's trying to figure out how to fix it. How can we prevent it?
Yeah, I think you'll hear from Dr. Dorsey this afternoon. He's going to suggest that if we can get some of the bad chemicals out of the environment, that we can prevent Parkinson's disease. Air pollution is a big one that's coming on, and maybe a cause of Parkinson's disease. There are unknowns such as the forever chemicals that are in everything, and TCE, which is a solvent, which is throughout the environment, and it's used particularly in dry cleaning. So banning certain chemicals is going to be extremely important. I think that we have to use common sense. Modern agriculture could not exist without pesticides and weed killers. We wouldn't be able to grow enough food to feed the country, let alone the world, without some of these chemicals. So we can't just randomly go and ban everything. We have to have common sense procedures and use of these compounds.
On that note, I have to say a massive thank you to you both. That was the most fascinating discussion. Am I right?